Assalamu alaikum, guys. Okay, so in this lecture, we are gonna talk about hemoglobin, which is a quaternary protein, the main oxygen carrier present inside the erythrocytes. We've talked about erythrocyte physiology in the previous lectures. I've given the link to the previous video in the comment section in the description, so please do check that out before viewing this lecture. Okay, now hemoglobin formation and its function, its structure, and everything. We are going to discuss that in this lecture. Now, everything related to hemoglobin in the red blood cells. Okay, hemoglobin is basically a pigmented protein which is present inside the red blood cells, the main carrier of oxygen in the red blood cells. If hemoglobin is not present in the red blood cells, then oxygen will not be transported in the blood. Okay, it has a quaternary structure because it has four polypeptide chains, four subunits, two alpha subunits and two beta subunits. Basically, two alpha and two beta chains, which each of the chain contain a heme group. So each particular chain contains one heme group. This makes a total of four heme groups to be which are present in one hemoglobin molecule. Heme group contains a ferrous ion. Basically, ferrous means an iron in the two positive oxidation state. So an iron ion in the two positive oxidation state, a ferrous ion coordinated with a porphyrin ring. Well, it's to be more specific, it's called protoporphyrin 9, protoporphyrin 9 ring, or it is also called tetrapyrrole ring. Okay, these rings are coordinated with the ferrous ion in the center to form your heme group, and each of the subunit, the alpha and the beta subunit, contain one heme group, making a total of four heme groups in the hemoglobin molecule. It contains a quaternary structure because it has more than one polypeptide chain. This is all the information that you guys already know. Now let's move a bit ahead. This is its structure over here. I've drawn each color represents one subunit. So two alpha subunits of the hemoglobin and two beta subunits of the hemoglobin, and they're present inside the red blood cell. And this is the major oxygen carrier. The iron has to be in the +2 stage to transport oxygen. Okay, if it's not in the +2 state and if it has been oxidized to the +3 stage, if the ferrous ion has been converted into ferric ion, then it was unable to transport oxygen. Okay, so it's important for the ferrous ion to stay as ferrous ion for oxygen transport. I've also drawn the heme group over here. This is how your heme group looks like, its chemical structure. Your iron or the ferrous ion over here in the center, and then it is coordinated with four pyrrole rings. This is one ring, this is the second, this is the third one, and this is the fourth one. So tetrapyrrole ring structure around the heme group coordination over here that forms your heme group. Okay, and this heme group it is made in the mitochondria, guys. This is important. It is made in the mitochondria of maturing RBCs. Acha, okay, so remember that the mature RBCs do not contain mitochondria. But uh, in the immature stages, in the newly formed RBCs, when they are actually maturing from the pro-erythroblast stage all the way to erythrocyte, well, we've done a lecture on the stages of maturation of red blood cells as well. I'll make sure to put a link to that as well in the description. Do check that out as well. This heme group, it develops in the mitochondria of the maturing RBCs. Okay, and okay, this was an important fact. After this, the hemoglobin formation begins at the proerythroblast stage and continues through the reticulocyte stage. These are the different stages of red blood cell formation or maturation, which we've talked about in the previous lectures. Even after the reticulocytes enter the blood, for 1 to 2 days, this heme group is synthesized uh, in the mitochondria of the maturing red blood cells, because even after the reticulocyte enter the blood circulation through the process of diapedesis, they are not fully matured, and their reticulocytes still synthesize hemoglobin inside of them until after 1 to 2 days the hemoglobin concentration reaches a maximum value. Okay, males have 15 grams of hemoglobin per 100 ml of blood, and females have 14 grams. And each hemoglobin can transport 1.34 each gram of hemoglobin. Okay, each gram of hemoglobin can transport 1.34 ml of oxygen, and each hemoglobin molecule can bind to four oxygen molecules. Okay, four oxygen molecules which make a total of eight oxygen atoms. 8 atoms of oxygen can attach to the hemoglobin molecule at a time. Okay, now let's look at the stages of its formation, the mechanism by which the hemoglobin is formed. This is a bit important, guys, so please do listen carefully. All right, so sorry. Okay, steps in hemoglobin formation. First of all, what happens is the two succinyl CoA molecules. These are the molecules that come from the Krebs cycle. Okay, these are made in the Krebs cycle. Krebs cycle. Okay, two succinyl coenzyme A molecules, they combine with two glycine molecules. Glycine is an amino acid containing hydrogen as the R group, and they both collectively form a pyrrole ring. Okay, pyrrole is a ring which is formed by two succinyl CoA and two glycine molecules. Okay, then four pyrrole rings, they combine to form a tetrapyrrole ring structure, or the protoporphyrin ring, which I was protoporphyrin 9, which I was talking about previously, protoporphyrin, right over here. I told you that the heme group contains a protoporphyrin ring, right? So for the heme group, this protoporphyrin ring is formed by a combination of four pyrrole rings. Okay, we'll look at this structure in a bit. Then this tetrapyrrole ring structure combines combines with an ferrous ion to form a heme group. Okay, this heme group is also called the prostatic group because it is a non-protein part of the this hemoglobin protein. Then the heme group, each heme group combines with one polypeptide chain, okay, which is known as the globin chain. Polypeptide chain, which combines with the heme to form the hemoglobin, is called the globin chain. So heme and globin, they both combine to form a hemoglobin subunit. Okay, this hemoglobin subunit, this is one hemoglobin subunit, one of the four polypeptide subunits of hemoglobin, either alpha or beta. It can be any of these two, okay, depending on the sequence of amino acids, depending on many other factors. And approximate MR of one hemoglobin chain is 16,000. So if I were to ask you the approximate MR of entire hemoglobin molecule, since we know that there are four polypeptide subunits, so the approximate MR of the entire hemoglobin molecule is going to be 16,000 multiplied by 4, which gives us an approximate answer of 64,000. So approximate MR of hemoglobin is 64,000. Anyways, now, two alpha subunits and two beta subunits containing their respective heme groups combine collectively to form the hemoglobin A. Hemoglobin A is the most abundant hemoglobin type found in the humans. Found in humans, okay, so this is how hemoglobin is formed. Okay, I've drawn a structure of a hemoglobin subunit right over here. I've taken this diagram from your book. You can refer to it from there as well. Uh, okay, zooming in. All right, so this is the structure. The ferrous ion coordinated with the tetrapyrrole ring, and each tetrapyrrole ring. Since this is one ferrous ion, one tetrapyrrole ring. So this means that it contains one heme group. This heme group has its own polypeptide chain. This polypeptide chain, the red dotted lines, they bind this heme group to a polypeptide chain. It can be alpha or beta. Okay. Uh, Right. So this is how the hemoglobin subunit is formed. And then in sickle cell anemia, okay, I've given a note on sickle cell anemia right over here. This is something you guys are pretty familiar with. In sickle cell anemia, what happens is that glutamic acid, which is a polar acidic amino acid, is replaced with valine, which is a non-polar amino acid, on the sixth position of beta chains, beta subunits of hemoglobin due to the point mutation on chromosome 11. Okay, and so the point mutation on chromosome 11. So when this type of hemoglobin, the sickle cell hemoglobin, is called HbS, is exposed to low oxygen concentration, it forms elongated crystals that are about 15 micrometers in length, which is basically greater than the length of a normal RBC, which is approximately 7.2 to 7.8 micrometers of RBC's length. So if they are much longer than that, the RBC concentration, RBC length, then they are more likely to rupture the cell membrane of red blood cells and make them more delicate, more fragile, which are likely to rupture the red blood cell membranes and makes it impossible for the red blood cells to pass through the blood capillaries, because again the blood capillaries are also much smaller than 15 micrometers in length. Okay, so this is a note over here. I'll make a very detailed video on this as well. Okay, guys, so thank you so much.